This paper is one of two, the third paper, that looked at this standard skin cancer model studied by scientists in the laboratory. It's done in mice. Cancer, fortunately, is a multi-step process. If cancer resulted from a single mutation, we would probably all be dead long ago. So cancer takes a series of mutations, a specific set of genes, to cause your cells to lose control. They lose the constraints. It's actually easy for a cell to eat food and divide. The hard part is for your body to keep your cells from doing that day in and day out. And that's a good thing, because if every cell in your body had unrestricted ability to divide and divide and divide, we would all fill this auditorium individually. So it's much more difficult to keep your cells in control than to let them just proliferate ad nauseum. And so cancer happens when those constraints are broken. Once a cell breaks those constraints, it is medically a cancer, and it will grow until it consumes you. This paper concluded overall, the induction of antioxidant enzymes by Protandim may serve as a practical and potent approach for cancer prevention. A follow-up paper, about a year later, looked further into the mechanism and showed two of the specific genes that are regulated by Protandim. Some genes are turned down, and P53 is one of them. And manganese SOD, that's a mitochondrial SOD, was induced, is turned up. And it was those modulations of those two genes may play an important role in the tumor suppressive activity of Protandim. What are the data shown in that paper? There were two groups of mice. The first group got a regular mouse chow diet. When they were treated with the two chemicals at high doses that cause skin cancer, that group of 15 mice, shown here in the yellow bar, got cancer. 100% of them, all 15 mice, developed cancers. Many of them developed, probably all of them developed more than one. The number of cancers per mouse, 6.3. The total number of cancers in that group of mice, 94. The second group of mice treated identically, except their mouse chow had Protandim added at a dose calculated to be equivalent to what most of you in this room are taking, 1 caplet a day for a human. 15 mice. This time, a third of them developed no tumors at all. And if you look at the total number of tumors in this group compared to the other, it's 57% lower, only 40 as opposed to 94. So the cancer incidence was reduced by 33%. The multiplicity, the number of individual cancers, reduced by 57%. This was a remarkable study described in these two papers. Paper number 6, published last year, Virginia Commonwealth University. A very complicated model. Protandim prevented fibrosis and capillary loss and preserved heart function, in this case, right ventricular function, right side of the heart. It prevented heart fibrosis. I told you Protandim turns down some genes and turns up others. There are genes that signal the fibrotic process, formation of scar tissue. And again, if you have a cut, it's important that you can heal that cut. But if your heart turns to scar tissue, you're in big, big trouble. The graph on the left shows the amount of fibrosis in the hearts of these animals. They were treated with, here, a chemical that caused pulmonary hypertension. Pulmonary hypertension puts a strain on the right side of the heart that pumps the blood to the lungs to get reoxygenated back to the heart, and the left side then pumps that reoxygenated blood to every part of your body. How do scientists measure fibrosis? The picture on the right shows a fibrotic heart, and you can see areas that stained kind of a bright blue scattered among the pink tissue of the heart. A normal heart is shown on the left, and you don't see those areas of blue. Look at the graph on the left. The control heart has very little fibrosis, that small blue bar. The hearts that had chemically induced pulmonary hypertension had about 4 times as much fibrosis, the red bar. Animals that were treated exactly the same way except with the addition of Protandim, and you can see the red bar was dramatically reduced, significantly reduced to the green bar. So fibrosis was prevented. We, in this case, measured a specific gene product. This is a protein that signals your body to create scar tissue. You can see in the control, the healthy animals, a very small bar. The ones with pulmonary hypertension, a lot of signal telling the cells to become fibrotic, to create scar tissue. And you can see it largely blocked in the animals that were treated with Protandim. Protandim prevented dilatation of the heart. What does that mean? That means enlarged heart. It's not good when your heart enlarges. You might think a big heart could pump more blood. Not so. It can't develop the pressure because the contractility is not there around that small space. The hearts from these animals, the control hearts, you can see that blue bar represents the diameter of the right ventricle, the pumping part of the heart that sends the blood to the lungs. And the ones that had pulmonary hypertension, the diameter was twice as great. The heart was enlarged to twice its normal size. And you can see a significant reduction in the Protandim-treated animals. If you look at the number of apoptotic cells, dead cells in these hearts, the healthy control animals had measurable apoptotic cells. The ones with pulmonary hypertension, many more times the dead cells. I forgot to put the green bar on that chart, but it's there. It's just reduced to 0. There weren't any dead cells in the animals that had Protandim supplemented diets. This is a measure of how well the heart pumps. It's cardiac output. It's blood, milliliters per minute of blood that heart pumps. And so if someone goes into heart failure, it means the pump isn't working. So a normal, healthy rat heart can pump 60 milliliters, a couple of ounces of blood every minute. The ones with pulmonary hypertension, that was reduced by about 40%. And in the Protandim-treated animals, you can see it's restored almost to the normal pulmonary cardiac output.